So good evening from India. Good morning to you. Hope you can hear me.
Is it audible to you? Yeah.
Yes, sir, you are.
Okay. Yeah, thank you, Priya.
So good evening, and hope you remember we have met once previously also. I'm lucky to have another session again, having you on chair and moderating it alongside Ritesh sir. Uh, at that time, it was a different entity and the program was different, but this time it's having the larger audience. We are having this via IFA, and IFA has now taken up this academic wing seriously, where we are not only building up the embryologists, but we are also empowering the others who want to have the free knowledge as and when required. And the wings are outreaching to every other embryologist in India. So it's pleasure to meet you again on online forum.
So just as a gesture, as a formality, although you, Dr. Carmen, doesn't require any introduction, but to brief about her. So Dr. Carmen, she is trained in science at University of Valencia, Spain. Dr. Carmen Rubio, she specialized in cytogenetic studies in human reproduction, and she has completed her PhD in the field of reproductive genetics and postdoctoral research, including research in male and female meiosis at laboratory of Doctors Patricia Hunt and Terry Haslord. So she has published more than 100 papers, and there are 229 publications at least to her credit, and 27,807 reads and 4,891 citations. Oh my God, well, that's commendable. So we are lucky to have you today as our guest speaker, and today she will be delivering her lecture on what's next in PGTA. So we have been doing PGTA. It's been over a decade now then that we have been doing aneuploidy screening. But now the world is moving towards non-invasive methods of doing aneuploidy screening. So what is the scope of doing non-invasive PGTA? And today, Carmen, she will be highlighting on that. So welcome, Dr. Carmen, and it's over to you, ma'am.
Okay, thank you very much. Thanks a lot for inviting me to participate in your webinar series. And I'm going to share my screen now. Okay, so you can see the presentation now?
It's upcoming.
Okay,
yes.
It takes some time from Spain to India.
Yeah, we can see now.
Okay, perfect. So, well, I'm going to cover during these 30-40 minutes what we have been doing in the last 6-7 years in the team here in Valencia. And as you mentioned, many of us, we have been working in the PGTA field for a long time. I started more than 30 years ago with the biopsies, and we are now at the point of considering this is the time to move to the non-invasive approach. And why moving to non-invasive approaches? We know that in other fields of medicine, we are really now at present working with non-invasive prenatal diagnosis and oncology. So for pregnant women, we can just have the blood and to identify the maternal cell-free DNA in the tube. And also in oncology, liquid biopsies are becoming the standard of care in some circumstances. So why not to move to non-invasive in the preimplantation embryo? If we consider the embryo as the patient, we don't want to be aggressive with the embryo. And having the embryo as a patient and the center of our investigation, we want to understand as much as possible from the embryos. We want to understand what makes an embryo the best to have a healthy baby at home. And in the current times, we have the opportunity of different technologies to gain this information and this knowledge from the embryo at different levels. The most common approaches now are the DNA, identifying chromosomal abnormalities. In the case of the culture media, is the cell-free DNA. And also we have a lot of information from imaging now because of the time-lapse system. What is the future bringing us? We still don't know. We will be able to identify many other markers of embryo viability, and the idea is just to combine and try to make our IVF treatment the most successful possible. So let's start what is the present now. We can identify DNA in the culture media, and we can test this with NGS. And this has been a fantastic discovery. We don't know why the embryos release DNA to the culture media when DNA is so valuable for an embryo and for life. But this is very, very, very clear, and we have shown that, and also others, that we can collect the culture media. We can identify these traces of cell-free DNA in the culture media. We can do the protocol to amplify this DNA. We can sequence, and then we can have nice sequencing plots, very similar to the plots that we are having when we biopsy 3-4 cells in a blastocyst. So this is becoming very, very, very interesting when we work with day 6 embryos. And let's go to what are the key questions that we have now related to the culture media. We still have many questions related to the origin and the composition of this cell-free DNA in the culture media. We need to understand if the results in the culture media are similar to the results of trophoctogen biopsies or the whole blastocyst. And we need to understand how comparable is the non-invasive PGTA to the PGTA to evaluate if this can substitute PGTA. So let's go little by little to cover these aspects. So starting with the origin and the composition of the cell-free DNA in the culture media, we know that the embryos release DNA along all the way in preimplantation development, but the truth is that we have enough DNA when we can culture the embryos from day 5 to day 6, and we have a blastocyst with a good number of cells participating in this process. There are several publications trying to quantify the cell-free DNA in the culture media. It's not easy because the amount is so low that the sensitivity of the measures sometimes give us bias. But what is very clear is that in media that has been in contact with embryos, we can identify an average amount of DNA of 10 to 20 picograms. So this is something between 1 to 3 cells. And in the control, culture drops without contact with embryos, there is no DNA. And this has been also shown in another study showing what is the concentration in picograms per microliter on day 4, day 5, and day 6. And you can see how the amount increases. And we consider that we are safe if we are analyzing the cell-free DNA from day 6 onwards, because the amount of DNA on day 4 or 5 is too low just to have proper NGS results. There are other studies trying to understand if this DNA in the culture media is coming only from the embryo or from another sources. We have to consider the potential contamination of the culture media with DNA from the embryologists or the technicians manipulating the aliquots of media, the dishes, but also we can have contamination of DNA from maternal cells for the cumulus cells that maybe only 1 or 2 are still attached to the zona and they can release DNA to the culture media or even floating in the culture media after denudation. That's why it's very important to start the process with properly decumulated embryos. Also, several studies using SNPs or by DNA methylation analysis have identified that the polar bodies can release DNA to the culture media, and that these polar bodies, if we go to day 6, then this DNA is more degraded. So the contribution of DNA from cumulus cells or from polar bodies on day 6 is lower, minimizing the risk of contamination. And this is what our studies and also this study of methylation had shown, that the contamination is higher on day 5 and it's better than to go to day 6 biopsies. So 2 reasons to go to day 6 blastocysts. The lower presence of contamination and the higher presence of real DNA from the embryo on day 6 to have a more accurate diagnosis. And we have been trying to understand a little about the preimplantation embryo dynamics. And if we can visualize this release of the cell-free DNA that you can imagine is really challenging. We started to work with Nicolas Placha, that is a famous cell biology researcher. He's now in the University of Pennsylvania, and we have been collaborating with him to try to do imaging in the final stages of embryo development using different dyes to stain the cytoskeleton and the cell nucleus. And with this, we have learned many different things, and we have seen a novel event in the latest stages of embryo development. We have seen that how like maybe 4 or 5 cells from each embryo start to have these small budding, these nuclear buddings that a little after they are detached from the nucleus. And maybe this is something that can start to explain how the DNA is released. But what is clear is that for sure this can have a connection with the origin of mosaicism in the human embryos. And this process of this budding of the DNA started to be higher when the blastocyst expands. That is when we have an cavitated blastocyst. And the hypothesis is because of the pressure of the blastocellic fluid, the cells are flattened in the trophectoderm, and they can have this pressure, maybe provoking this release of the cell-free DNA. And this is the studies that we have published recently in Cell, and we are still working on this. Let's move now to other different studies, more clinical studies that has been performed, trying to find the concordance rates of the cell-free DNA in the culture media with the results of the corresponding trophectoderm biopsies or even the whole blastocyst. There has been, as you can see, a list of studies published so far, and it's really great to see that it's not only us that we have observed this phenomenon. And I have highlighted here the results in which day 5 and day 6 were compared, and you can see how informativity, concordance, and the accuracy parameters are higher when day 6 analysis is performed. In overall, we can say that the informativity rates on day 6 can be close to 98% if we have good quality blastocysts, and deployed concordance with trophectoderm biopsies on day 6 can be now close to 89%. We consider that our standards are having informativity rates around 90–95%, and the concordance rate when we are doing the validations with the clinic should be above 80. But said that, we consider now that working in the optimal conditions is giving us concordance rates of 85 or 90%. That you will see later on, this is very similar to the concordance rate with the trophectoderm biopsies and the inner cell mass. So let's see 2 specific studies that has been published comparing the non-invasive PGTA with the PGTA. This is a study published or commented by Catherine Rakowski in a News and Views journal in fertility and sterility. And what Catherine Rakowski was presented is that she was comparing the results of culture media and trophectoderm biopsies with the results of the full blastocyst in a number of blastocysts. And what she observed is that the sensitivity and the specificity for the non-invasive was mainly the specificity was higher for the non-invasive, and in overall the concordance was 94% of the media with the blastocyst compared to 85 of the trophectoderm with the blastocyst. So she was observing nice results in the culture media. And also in this second study by a Japanese team with only 10 blastocysts that they were growing until day 10 or day 12, they were comparing the sensitivity and specificity of the non-invasive in orange compared to PGTA in blue, and they observed better results again with the non-invasive. So we have some clues that the culture media, when collected and when the embryos are properly handled in the IVF lab, can give us very accurate results. And now I'm presenting one of our biggest studies performed so far. So we started with this, as I mentioned, more than 6 years ago. We did different pilot and proof of concept studies, and we have just finished a prospective multicenter concordance study, including more than 2,000 samples. In total, 2,538 blastocysts, in which we had trophectoderm biopsies and culture media. And in a subset of these blastocysts, we were able also to have results from the inner cell mass. So we published the interim analysis 3 years ago with 1,300 blastocysts. And now we have collected the results of the final study. We are working in the submission for publication, but I can advance you that our concordance rate of the cell-free DNA with the trophectoderm biopsies was in overall 78%, with a range between 73 to 83, but most of the centers, they have more than 80%. And when we have samples from both, from the trophectoderm biopsies, the inner cell mass, and the culture media, we observed that the concordance rate of the cell-free DNA with the inner cell mass was 87%, and of the trophectoderm biopsy with the inner cell mass, 90%. So very close concordance rates of cell-free DNA and trophectoderm biopsies with inner cell mass. So we are really very close to a trophectoderm biopsy. What else have we learned in this study? We have learned that if we are working with a benchtop incubator, because in this study, all samples were incubated in regular benchtops incubator, 5 different models, the results were very similar. Also, we were working IVF centers, more than 10 centers were working with different culture media and combinations with different incubators, and very reliable results. And this is very important because with other methodologies as metabolomics, there is a strong dependence on the culture conditions with the results. But with DNA, the DNA is very robust. So DNA works in different culture conditions and different situations. What else have we learned in this study? So we have been able to collect results for 304 single embryo transfers. In these cases, embryo transfer was always performed according to the trophectoderm biopsy, because in this study, all patients undergoing PGTA, embryos were transferred according to the results of the biopsy, and in parallel, we were collecting the culture media blindly to do the concordance study. So after transfer of the embryos, we were able to pair the clinical outcomes with the results of the culture media. And we observed that in those cases in which both the trophectoderm biopsy and the culture media were euploid, we had a lower miscarriage rate, 14% compared to 25% when the culture media was aneuploid. So if the culture media is euploid and also the trophectoderm biopsy, then we have lower miscarriage rate after PGTA. And if we consider the main outcome, that is to have ongoing pregnancy rate, we observe again that if we have a euploid culture media, the final ongoing pregnancy rate is higher, even though in every case we have a euploid trophectoderm biopsy. So imagine in cases without a euploid trophectoderm biopsy, this difference can be even higher. So these results are very supportive, and the sample size now, as I mentioned here, is 304 single embryo transfers. And in our pilot study that we performed in 2019 with just a single center in Italy, was showing also this, that miscarriage rates, if we have aneuploid aneuploid cell-free DNA, are very high, even though we have a trophectoderm biopsy. So we are observing the value of the analysis of the culture media as a representation of the full blastocyst. And regarding the comparison with the inner cell mass, we did this comparison with 2 centers, Boston IVF and Nilo, France. And with one of these centers, with Boston IVF, we were able also to do a parallel study. We wanted to see the impact of extending culture in all embryos to day 6, even though if we have nice blastocysts on day 5. And with the support of Dennis Sakas from Boston IVF, we were comparing the results of the patients that were participating in this study, the multicenter study, in which one of the importance was to push culture to day 6 to all patients included in the study. And then we were comparing the results with the regular IVF program, in which sometimes they did the biopsies on day 5 and other times on day 6. And you can see how regular PGTA cycles and the study cycles, similar euploidy rates, similar miscarriage rate, and similar live birth rate. So the fact that we need to do extended culture to day 6 in every case for the non-invasive PGTA is not decreasing live birth rates. You can see here in orange, live birth rates were very good and similar to those cases in which PGTA was performed either on day 5 or day 6. And there are publications related to this, that if we are doing a chromosomal analysis in the embryo, then the day of vitrification or the day of transfer is not having an important impact in outcomes. It's different if we are working with embryos without a previous chromosomal testing. So this is evidence to support that extending culture to day 6 is safe for your patients. And with all this experience that we have been collecting after the application of this non-invasive PGTA test to patients, and with the knowledge from different studies, how can we move to clinical application? What should we consider is key to have successful results? Very clearly, to be aware of the risk of contamination from cumulus cells. So we need to minimize the presence of contamination. Secondly, we need to incorporate some modifications in the lab protocol to be able to have the higher amount of DNA and the minimal amount of DNA from contamination, from external or maternal contamination. Timing for media collection is going to be key, as I have already mentioned. And then how we analyze the results of sequencing. We cannot apply the same ideas that we apply to the analysis of trophectomy biopsies. This is completely different, as you will see. So let's start with the key part of contamination and the IVF lab protocol. We need to be aware of that we have risk of this maternal contamination from cumulus cells or polar bodies, external from manipulation of the culture media, and also from contamination in our working spaces, the hood, our hands, everything is very important. That's why how can we minimize contamination? Like very basic steps, common sense steps. During embryo culture, how can we decrease maternal contamination? Very clearly, we are going to collect culture media only for the latest stages of embryo development, only from day 4 to day 6. So on day 4, it's very important to do as an extra step of washing the embryos individually to remove DNA from the previous stages, to remove DNA from polar bodies, from necrotic cells, from cumulus cells. At this point, if there is still a cell attached to the zona, you can detach it very easily. Then transfer the embryos to a fresh drop of culture media and then culture until day 6. With this, you can really minimize to 0 the presence of contamination. And then from our side, from the embryology side, it's very important just to prepare the area in which we are going to work, to clean very well the working spaces, and also to protect ourselves not to release a DNA, not to release cells to the environment by covering properly, like with caps, with masks, with gloves, but also the arms. It's very important to have like a surgical gown and to cover everything, because our arms are inside the hood, are in the environment that surrounds the space in which we manipulate the DNA. So this key and basic steps can really result in very good results. So this is an overview of the protocol that we are working with. We are having very nice results when we combine the embrace with the embryoscope. When we need to culture embryos individually from day 4 to day 6, very important. If you have a time-lapse system or embryoscope system from day 4 to day 6, you need to culture individually. Also, if you work in regular culture dishes or regular incubators. So very important after the nudation, just after your regular washes in the nudation, to add an extra additional steps of individual washes. You can use this kind of dishes that they have multi-wells with 25, 30 microliters, and you can do 4, 6 washes in these wells. And then you can place in culture until day 4. On day 4, very important. This is for us the really key step, day 4. Then one by one, embryo by embryo, you should wash each embryo in a different row of culture medium and use an individual capillary for each embryo. And then you can place in the fresh culture dish and wait until day 6 to collect the media individually again with individual tips and to place in the tubes and to freeze immediately at -20, preventing the release of oil, because oil will prevent the proper NGS amplification, and then also preventing shaking the media and strong changes in the physical conditions that can fragment the DNA. So very simple. Once you get practice, it's not incorporating too much complexity to the IVF lab. And regarding the timing for media collection, I have already presented you the list of papers doing this. We learned about this very early in the 90s, in 2019, sorry, because we were doing this pilot study in collaboration with Laura Girardi and Filippo Ubaldi in Italy, comparing day 5 results with day 6. And we observed how informativity and concordance rate really were much, much higher when we were collecting samples from day 6. And this is for fresh cycles. That means that we have the blastocyst that has not been thawed before. And we have done validations with many centers all around the world. And now, with these validations, we can see that in fresh cycles on day 6, we can have concordance rates of 86% with conventional incubators. And if we move to time lapse, our concordance rates can be even higher, close to 90%. So very nice results with fresh blastocysts. But we have also many studies published in which the concordance rate has been calculated with donated embryos for research that were frozen. So you can thaw the blastocysts and keep in culture for some time, and then the release of DNA is even higher. We have done one study showing that if we have day 5 blastocysts and we culture 8 hours, then we have low informativity rate. This is the blue bar. Only 8 hours on day 5 blastocysts, low informativity rate. But if we thaw a blastocyst on day 5 and we keep in culture overnight, then we have very good informativity rates, very similar to the informativity rates of the whole blastocyst. And then with day 6 blastocysts, that they have higher number of cells in average, you can thaw in the morning, you can collect the media in the afternoon, and you can have really high informativity rates. And the concordance rate, as you can see here, for frozen blastocysts are always about 90%. So very good results in previously frozen blastocysts. And these are the results of some of our validations with frozen blastocysts, in which you can see that either conventional or time lapse, again, concordance rate are very good. So this is related to the protocol in the IVF lab and the timing for media collection. But what about the genetic analysis and the genetic testing? I have to say that the protocols for NGS in the IVF lab, they always have a step of whole genome amplification. And the way in which we amplify the DNA from the culture media has been modified a little to be able to get the best from these small concentrations of DNA in the culture media. So we have modified the whole genome amplification protocol for culture media to improve the results. And also it's very important to identify how to diagnose it. Once we have the NGS results from the sequencer, we should apply different criteria, as I mentioned, to the trophectoden biopsies. And there are two studies showing this. This is one study also performed in Boston with Catherine Rakowski, in which Catherine observed that to report the results in the culture media, any aneuploidy below about 50%, 50-60%, if we report aneuploidies at the level of what we would consider low mosaicism, then we are going to have a huge percentage of false positive. We need to understand that the results in the culture media are noisier, and the noise can be misdiagnosed as mosaicism because of the nature of the DNA. So we should not report mosaicism in the culture media below 50%, because most of the cases, this is going to be a false positive, and then you are going to have a result of an embryo not to transfer. So below 50%, a lot of false positive rate, we should only report aneuploidies above 50%. And this has been shown also in another study by a Chinese team, showing that the concordance rate, when you consider 50% as the threshold for aneuploid, is 87. And if you put the threshold at 20% or 30%, then only concordance rate is going to be only 74%. So very importantly, you need to consider the threshold to call a culture media as aneuploid about 50%, not consider low degree mosaicism. We have been working in this sense just to have a specific cutoff values for the culture media for each chromosome, different from the ones that we are using for trophectoden biopsy. And with this, we have identified the results in the culture media and the comparison with the trophectoden biopsy, and we have established for different results in the culture media the probability of corresponding to a euploid blastocyst. And we are able to prioritize the embryos for transfer. So you can consider the culture media with two applications to say that an embryo is euploid or not, as in PGTA, but we recommend to consider this as a prioritization, as it is occurring also in PGTA. So we have embryos that has a higher priority to be normal, an embryo that have a lower probability of being euploid. So we can just rank them and give you a ranking to identify which embryos you can transfer first. And with this theory and with these suggestions, we have many centers all around the world working with non-invasive PGTA, and I'm glad to say that last year it was really rewarding just to collect experience from many of them in the first Embrace user meeting that we have at ERRE last year, before ERRE. And I'm going to show you some of the results that your colleagues were sharing with us, other IVF centers using a non-invasive PGTA. In different senses. I'm showing here results, for example, from Ruber International, that is a Spanish center, applying this non-invasive only in young patients. They are doing PGTA in patients above 38, but in young patients, they are doing the non-invasive for any IVF patients with good prognosis. And they are comparing the results with the morphology, and they are observing that if they apply non-invasive, they can increase their ongoing pregnancy rates to 75% compared to 55% with just morphology in good prognosis patients. Another study was presented last year from IVF centers in Latin America, and they observed very similar things, that just with morphology, elective frozen embryo transfer with morphology, they have ongoing pregnancy rates in general, an indication of 26.6. When they compare with Embrace, the percentage increased 10 points to 36%. Also, in this center from Taiwan, they were doing a kind of non-selection study. And then after doing the non-selection study, they identified that when they were transferring euploid media, they have a very good live birth rate. And when they were transferring blinded because it was a non-selection study and employed culture media, they didn't have a pregnancy. So another suggestion that it was working nicely. Another center from different country, Austria, they were applying this test in patients with good prognosis, recurrent miscarriage, recurrent implantation failure, poor blastocyst quality, or advanced maternal age. And they have, in these poor prognosis patients, they can have 45% of ongoing pregnancy rates. Another team showing results from Dubai. Here again, they were showing results with embryos in patients with more, this is only patients with about 38 years of age. Again, 46% pregnancy rates with non-invasive compared to 35 with morphology, 10 points difference. So this is showing that consistently different centers, different countries, different indications, you can improve your ongoing pregnancy rates. And in this group, this is a group in Poland that they are really very active with Embrace. And they are doing Embrace in a good percentage of patients. Indeed, a percentage of their patients doing PGTA are now shifting to Embrace. And they observe similar blastulation rates in Embrace and in PGTA, similar percentage of good quality embryos, and their clinical pregnancy rates of Embrace was very similar to PGTA. And because of this, now they are trying to avoid biopsy because they are having very good results with Embrace. And what they were sharing with us was the patient's decision in many cases to go to the non-invasive. So it was, this shift to Embrace was also driven by patients' opinion. So in conclusion, what I was trying to show you today here is that non-invasive PGTA with the analysis of the embryo cell-free DNA is becoming a powerful biomarker to assess embryo viability, considering embryo viability as the chromosomal status of the embryo. That this can help us to prioritize the embryos with higher probabilities of being euploid and resulting in a healthy baby, and this will shorten the time to pregnancy and the need of unsuccessful transfers before having the right embryo. And this can be a very interesting option also for patients that they don't feel comfortable with the biopsy or discarding embryos. So just prioritize embryos, but without discarding them for these patients with these maybe ethical or religious concerns. And then a summary of the user's experience is that we can have clinical results very similar to PGTA and better than morphological embryo selection. And this can be observed for different indications. So this is just my final slide. I would like to thank, first of all IVF centers that has been collaborating with us. We are having close relationships with IVF centers. This is our aim, just to learn together. I would like to thank all the R&D team that we have in Valencia with a strong importance of the bioinformatician team. And this is the Embrace team that they have been the team really working very, very, very close with this. So it's Nana, Damia, and Luis that is now the head of the department, and Lucia, our technician. And as you know, we have been collaborating from the very beginning with Carlos Simon. Now Carlos Simon is not part of VitroLife team, but is part of Carlos Simon Foundation, and we work very close together. He's in the building just nearby. And also we have a very nice collaboration with Nicolas Placha team. And from VitroLife, we have Ana Domingo as a postdoc in Nicolas Placha laboratory for these basic research studies. And thank you very much for your attention.
Great, thank you very much for that wonderful lecture. Obviously, there have been a lot of questions with regards to this being a new technology on is it really worth? Why day 5? Why day 6? Why not day 5? Why cell-free DNA? Obviously, the question and answer session now is open. So all the people, all the audience out over here, those who want to put in your questions, you can put it on your chat box or you can raise your hand and we can pick you up from there. But till that, thank you very much. we know that technology has been moving at multiple folds. It was just around the clock that PGTA was accepted, and the NIPGTA is now clocking the talks. And at the same time, it's really increasing at a very fast pace. And we still are actually wondering how it is. And I think your lecture has thrown upon some good insights on how people around the globe are using NIPGT, and it's been well accepted overall. So thanks for that wonderful session. The session remains open for question and answers.
Yeah,
Till that time,
definitely.
sir, please,
That
sir.
was really wonderful talk, and I think you have covered most of the topics regarding the protocols, regarding the methodology, regarding the sensitivity and specificity between the PGTA with using the biopsy invasive procedure versus non-invasive procedures. By the time we start getting the questions, just a very basic question I would want to ask. So first, it is a very basic thing. So what is the indications for non-invasive PGTA? Although they are more or less the same as those for the PGTA, but I would want to ask you, so what are the indications when the clinicians should request for this investigation?
Okay, this is a very, very good question and has not an easy answer. So let's explain you. I was trying to show in the presentation how the test is applied differently in different countries and in different scenarios. So I will try to cover this. So if you are a country in which PGTA is extended and is allowed, I think that advanced maternal age patients, they feel comfortable, for example, having a PGTA because they want to minimize the risk of aneuploidy and the huge aneuploidy rates related to age. So I think that if you are in this scenario, most of your patients, advanced maternal age patients, are going to prefer PGTA. But said that, this was also my first thought. But said that, we have learned that even some patients of advanced maternal age, that they know that they have a high risk of aneuploidy, they don't feel comfortable with the biopsy, that they prefer not to touch the embryo. And in these cases, you can do the embrace. So I think that the very important thing with embrace is to understand the patient. So today, advanced maternal age, gold standard, PGTA, trophectomy biopsy. But you are going to face this situation because many of our clinics have been telling this, that patients are starting to read about non-invasive and they feel more comfortable not doing the biopsy. Then this is the case. Or also if they have only 1 or 2 embryos, the quality is so-so, and you don't feel comfortable doing the biopsy. So this is for advanced maternal age. For patients that are younger, I think that the non-invasive makes a lot of sense. I think that for younger patients, recurrent implantation failure, recurrent miscarriage, good prognosis patients, I think that in these cases, it makes a lot of sense to move to the non-invasive.
Okay, that really actually makes sense. And actually, these are the patient's indications where with the patient or couple is with advanced maternal age, where the female is more than 35 years of age, and then with a history of recurrent miscarriage or implantation failure. So basically, those are the cases where this is indicated. And as you said, yeah, Carmen, in India, we definitely, as per new ART guidelines, we have been advised to stick to these 3 parameters. So only request for PGTA or NIPGTA, where these are the only in the indicated cases, and it should not be a a screening procedure. It is not a screening investigation, and it should not be offered to all the patients or the couples. So
Okay. I think this, as any new technique, when you start, you need to be conservative, to be cautious. But with this, what you will see gaining experience is that this can improve your regular good prognosis patient results. But I agree that we should go step by step.
Right.
So another very basic question. Though we have discussed the presentation in length, but most of the embryologists still worry about what is the origin of cell-free DNA and how is it bypassing the zona of an embryo or oocyte into the culture media. So what's your take on it?
Okay, I'm also very curious about this. So we are really curious. We need to understand different aspects. So we need to understand if the cell-free DNA comes mostly from the inner cell mass cells, trophectoden cells, or both. Our current experience is that it should come from both. So it makes sense. Then what are the molecular mechanisms making these fragments of the cell-free DNA? Is it small fragments from many cells? Is this DNA from few cells that explode and release the DNA? This is what we are trying to discover with Nicolas Placha imaging research. And then we are trying to visualize the moment in which the cell-free DNA goes externally from the cells to the perivitellin space and from the perivitellin space to the zona. But so far we have not been lucky because all the dyes that we are using, they have a lot of noise. So if you try to go to the this level of resolution, you have a lot of noise. So we are just starting to understand cell dynamics that can explain where the DNA comes from. And this is the paper we have in Cell. I cannot disclose some of the latest things that we have seen imaging videos, but we are observing another phenomenon in the blastocyst that would make a lot of sense, explaining how this nucleus can really fragment it. I think that my feeling now is that some cells in the embryo, they fragmented the DNA in a non-apoptosis way because apoptotic markers are negative, and these fragments are going to be released. And the amount of cells that are suffering this is about 4-6 cells at extended blastocyst. So then it really is aligned with the results that we are observing. We are observing DNA corresponding from 2 to 4-6 cells, and we are observing DNA in expanded blastocyst. That is when this phenomenon starts to appear. So, but still many questions.
Yeah, because you did also mention about nuclear budding, and that's something which is new, I suppose, for a lot of people, because the initial thought was that obviously, as you also mentioned, that there is apoptosis happening, there are certain cells getting degenerated, they release the DNA because they are getting degenerated, and that comes out in the culture media. And then do that become the representative of the embryo where we are actually taking up for NIPGT. So that was the earlier thought, which your presentation showed that there is a nuclear budding and that might come out, and that is also the source.
Yeah, we are not 100% sure what is the origin, but we have tested that is not apoptotic DNA. And just to be sure that we minimize the risk of having apoptotic or at least DNA from classic apoptosis, then it's very important this day for washes, because we know that the embryos can start fragmentation day 2, day 3, mainly because of necrosis, but maybe some apoptosis, but they don't have so strong fragmentation after, once you have a blastocyst or a morula, you don't have so active fragmentation process. So by collecting the media from day 4 onwards, we also minimize the risk of taking DNA from the embryo from necrotic events. This is at least the theory.
Okay. Would definitely be a fascinating paper if we are explaining to everyone how it travels out from the zona and coming into the media and what is the origin. I think that will answer a lot of questions, and that will throw up a lot of light on NIPHT on why it is giving so nice concordance these days with PHT.
Sure. We need more basic research study to understand it. Sure.
Right. I think we can move on to the question. We have a couple of questions here. So first is that, do the results of NIPHT get affected due to the frozen versus fresh embryo spent culture media collection? So I think they need to ask that what is the difference between going for NIPGTA with the frozen versus fresh embryo transfer, because that's the limitation with the frozen embryos, I think. So what's your take on that?
Okay, well, I need to clarify this. When we say NAPT, NAPGTA with fresh, we collect the culture media from fresh blastocysts, but we need to vitrify the blastocysts to do the analysis and to do a deferred transfer as in PGTA. But we are collecting the media from a fresh blastocyst. And this is the typical results that I have shown you. But we have had the need to test frozen blastocysts for patients that had the cycle and they didn't get pregnant or had a miscarriage and they wanted to test the surplus embryo. So if we have a frozen blastocyst, we have done the study on, because we already have a blastocyst, how long we should culture, because we cannot culture 48 hours as in the fresh ones. We need to culture shorter term. And we wanted to identify the shorter that we need to culture, not to expand too much the blastocyst expansion. So with day 5, we need overnight. And with day 6, only 6 to 8 hours. And why with day 6, with only 6 to 8 hours, it works nicely? This is not the same for fresh. We think that the release of cell-free DNA from frozen blastocysts is more active. At some point, vitrification should be impacting, as previously described, the fragmentation of DNA. And at some point, we know that vitrification is impacting the texture of the zona pellucida. And we think also that there is something very interesting because of the vitrification process. So we vitrify the embryos and we thaw them. When we are thawing them, all the thawing steps are allowing us to clean very well the embryo from other sources of DNA. That's why I think also it's very accurate, because no external DNA. And also during the expansion process, after thawing, maybe this expansion process is releasing more DNA to the culture media. But again, just theories. But it's very consistent in different studies that the higher concordance rates are always with the previously frozen blastocysts. So something is there, and we are trying to justify this. But what is the risk of working with frozen blastocysts? We cannot apply this as a general practice, because that implies for clinical applications 2 vitrification steps. So this is only for specific cases, but sometimes it's allowing you to rescue. And you know, some centers now that when they have non-informative results in PGTA, they thaw the embryos, they culture, and we collect the culture media. So we don't need to do a second biopsy. You can just collect the media. So you are going to have 2 vitrification steps, but only 1 biopsy. So these are some of the applications that some clinics are using. Different clinics have different ideas on how to work with this.
That's really very interesting. Actually, in fact, I was also in an opinion that maybe the fresh culture media for NIPGTA, fresh culture media is the best way, and for the frozen ones. So, but there is something because of the shrinking of the embryo and re-expansion. So, zona pellucida that develops some ability to discharge more of the inner cell thing. SBM, I think. So that is you mean to say.
So maybe when it is frozen and thawed, while it is re-expanding, is there more of a nuclear budding?
And many times you also answered another question, because I was going to ask. Many of my doctors, they often ask me if during the first instance, at times it happens that you get no DNA reported, right? So they are quite apprehensive that in the first instance we are not getting any, the DNA is not been detected, and now they don't want to rely on NIPGTA, so rather they want to jump to the invasive procedure. So
okay. If you have the analysis of the culture media and it's not invasive, you have this option to do the biopsy to be reassured. But what we are applying is what we call, instead of rebiopsy, for PGTA, we are applying the term of remedia. And what we are doing, and is being very successful, is if you have a non-informative culture media, you can thaw and you can culture 6 hours, 8 hours, the medium, the embryo, and you collect the medium a second time and you rescue it. If the blastocyst is properly expanded, you rescue it in a similar way in which you rescue with a biopsy.
Okay, right. So this is a good tip. And next, the continuation of the same question is that normally morula stage embryos are said to be susceptible to external factors. Isn't that the case for embryos, as you suggest culturing from day 4 to day 6 instead of day 3 to day 6?
Well, at morula stage, embryos are very robust. So washes, manipulation at morula are very versatile. So washes on day 4 are working nicely. We have been talking with the companies that are providing the culture media, and now with VitroLife, you know now that we are part of VitroLife team. And we have been talking with them about delaying, if you are using sequential media, about delaying the media change from day 3 to day 4. There is no issue. They have tested it. And extended culture from day 4 to day 6 or day 7, no issues with the culture media. Embryos are more tolerant as they age in the lab. So the oocytes are they're really very sensible cells, we know, for temperature, pH, osmolarity. And as we move, as we advance in embryo development, the tolerance is higher in the embryo. So morula are robust as well as blastocysts, as we know. Blastocysts tolerate the biopsy. So we know they are really has a high tolerance.
Okay. So that's there is one question. I think that's from Dr. Harita, and she says that it's insightful talk. Dr. Carmen, thank you. And her question is that in the table showing studies from the last few years, the method of WG8 technology used were different. Impact of different technologies on NIPGTA results. So so what she's asking, what's the impact of different technologies on NIPGTA results?
Okay. So I guess that the person that is asking knows a little about molecular biology. Okay, I will say we have tested many different amplification protocols. So talking about amplification protocols, we need to identify the enzymes that do the amplification and the specific steps of the protocol. So talking about the enzymes and about the type of amplification, regular MDA, multiple displacement amplification protocols, are not working nicely with culture media. PCR-based protocols are working nicely, as the ones based in Sureplex with arrays, the ones with Thermo Fishers, the ones with VeriSeq, the ones with EmbryoMap. All of these are PCR-based, and they are working nicely in the culture media. The standard MDA ones that they are mostly used for karyomapping or for monogenics, they are not the more regular one for aneuploidy. These are not working with fragmented DNA. The standard ones, you can modify them, but they are not the best option. So in general, the protocols that work nicely for PGTA, the enzymes should work nicely for the culture media, but you need to change the conditions during the protocol, but just with the same enzymes.
For that. And the continuation of her question is, how different is the NIPGTA algorithm from NGS? Trophectoderm biopsy PGTA, and how does the noise makes it challenging for the reading results?
Okay. Well, I will need 5 minutes to explain this, and I will explain a little how we develop the algorithm for PGTA and how we develop the algorithm for the culture media. So for PGTA, the way in which we are using a commercial platform for Thermo Fisher, They have their own software, but every time we incorporate something new, we do our own validations that we publish. So we have cell lines with different aneuploidies for different chromosomes and also for segmental aneuploidies. Also, we prepare mosaicism-like models to evaluate at which level of mosaicism we can detect properly. And then we establish the thresholds to detect mosaicism, for example. So we do mixtures with different proportions of DNA to identify if we want to detect 30% of mosaicism. It's not just 30 going to the software. It's a different value. Maybe it's 32, 35. So for each chromosome, we identify when to establish the threshold to call mosaicism at different levels. And also we establish the minimal size of a segmental aneuploidy that we can detect without overdiagnosing that. So this is how we work with PGTA. Cell lines, different abnormalities, and models for mosaicism. How are we working with non-invasive? With non-invasive, we have done this big study, 2,500 embryos. We have trophectomy biopsies and culture media. And in about 300 cases, we have inner cell mass. So we were comparing the results of the culture media with the results of the trophectomy biopsies. For each chromosome, one by one, chromosome 1, chromosome 2, we were identifying the threshold that allows to have higher concordance rate of the medium with the trophectomy biopsy. So, and this is how we have identified chromosome 1. Everything below 50%, we should report as normal. Everything above 50%, we should report as abnormal. Chromosome 2, imagine, everything below 48% or 52. So chromosome by chromosome, we have been trying to see when we have the right threshold. It would be fantastic to do this with trophectomy also, to have several trophectomy biopsies to identify when we have the higher concordance. Nobody was doing this with trophectomy biopsies. We have few cases comparing with inner cell mass, but this is the way. That's why it's different. With the culture media, we cannot use cell lines. We cannot use cells to develop an algorithm. We are using the culture media by comparison with the trophectomy biopsy, and this is what is different.
Well, that's something which is throwing light at the details, because we didn't knew about this intrinsic challenges possibly which you all must be facing, which I think people would not be aware of how it was established. Obviously, this questions of details with regards to a bit of a molecular biology has given us a new insight that how you have changed it with regards to chromosome to come to one conclusion, and then that is being reported by whosoever the geneticist or is there in the lab, looking at all of them and then asking it, reporting it as mosaic, low mosaic, high mosaic, euploid or euploid. Thank you for that detailed explanation.
Yeah, and if I can add something, because we are facing every day, you have started with day 3 biopsies, and I guess you have moved to trophectomy biopsy. So if any of you is familiar with the NGS profiles with day 3 and trophectoderm, you have seen how day 3 they are noisier and trophectoderm should be flatter. Imagine that you want to diagnose mosaicism in a day 3 profile. You are going to have a lot of low mosaics because by nature, the day 3 biopsy is noisier. This can happen with the culture media. So you have to bear in mind that the culture media is more similar in the profile to a day 3 biopsy. So it's easier to apply the mind of the day 3 to the media because this noise is not real. Trophectomy biopsy, you have good cells, it's flattened. You can trust on mosaicism at a certain level. Again, if you have a trophectomy biopsy with multiple mosaics, do not believe this is noise. So I have seen reports from non-invasive saying all the embryos are abnormal mosaics. And I say, no, what a pity. You are discarding embryos. When you have a noise in the culture media, this is nature. Just think day 3 biopsies. How you will interpret this?
Why that is noisier? Maybe anything to do with the cumulus cells, maybe because of the noise of the cumulus cells. Why I did not get that thing. So why are
Well,
day 3 more noisier?
Yeah, the noise is related to the amount of DNA. In a day 3 biopsy, you have 7 picograms. In a trophectoderm biopsy, depending on the number of cells, you have 30, 50 picograms.
Okay.
In the culture media, depending on the volume and how you grow, you can have a variable amount of DNA. You can have from 7, 10 picograms to 30. But if you are in the lower range, then you can have in some embryos noisy results.
Because the DNA is less, that's why. So it's more noisy. Yes, yes. Right.
And mainly if you go to day 5, if you collect medium from day 5, you have very low concentrations of DNA, sometimes lower than the amount of DNA that you have to expect in just one single cell. So then on day 5, you have a lot of noise.
Okay, the lower the cells, the more noise.
Exactly.
Next part of the question is, yes, I think most of the embryologists, they would want to ask, so is there any impact of assisted hatching on collection of the cellfree?
Okay, we have done, well, for sure more clinics have tested that, but we have knowledge of 2 or 3 clinics that they wanted to test this. So when they were doing the validations, in some embryos, they were doing assisted hatching on day 3, and in another embryos, they were not doing assisted hatching, and the results were very similar. The fact of doing assisted hatching on day 3 doesn't make impact at all. What I can say is that some IVF centers, I don't know what is your practice there, your regular practice, for frozen transfers, when they thaw the blastocyst and before transfer, they do hatching of a big hole. And if this is the regular practice of your center, just apply this also to the embrace. So if after thawing you do a assisted hatching before transfer, that is okay. This is what we have observed in some centers, that at the very beginning they were not doing this because of embrace, but they were doing in other patients. And when they started to incorporate this regular practice, they started to have higher pregnancy rates. But again, this is, some centers have adopted this practice and other centers don't. So it depends on what is your experience on that. Assisted hatching before collecting the media, no need.
There's no need.
There's no need, yeah. And as you said that even the frozen cells, more or less, the frozen embryos, they have a special, the zona is has a tendency to share more cell-free DNA to the surrounding culture media. So especially those cases, there is no need.
No need. No need.
And final question is that the experience from India Igenomix lab,
is it different from your parts of the world, or is it different? India versus Europe or otherwise, like experience from India lab of non-invasive PGTA.
Okay, I don't have, I have not prepared results, but one very, very important thing when you are starting either PGTA and embrace or non-invasive PGTA is to have a good culture system in your lab. You need to have a good culture system that support embryos until day 6. So if you have a good culture system that support embryos until day 6, you can have similar results to the results everywhere in the world. What is very important, and we always say, is to follow strictly the protocol. We always say, please follow strictly the protocol. And you are embryologists, I guess all of you all suffer the pressure of having a lot of work and maybe not enough staff in the lab. And sometimes during the non-invasive PGTA protocol, you have to take some more time to do the washes properly, to dress properly, to clean the surfaces. So please do not skip any step. Even if you think it's a silly step, don't skip any step, because these little things make it work. So I will say that the very important thing is to follow strictly the protocol. It works.
Okay, so it is critical that you follow the protocol as precise way as possible, exclusively because NIPHTA is also right now we know that there are noise which are generally reported even with small amount of anything which comes into the media. And as we are looking into media, as you already said, we have low amount of DNA and it creates a lot of noise, and with some or other contamination, it will keep increasing and make a life of geneticists difficult to report them. Right? So
yeah, so proceeding to the next question. So we have a few questions. How does the use of NIPGT for the nuclear transfer of embryos can be done?
Oof, what a question.
yeah.
Okay, okay, well, I don't know how to apply it. I was approached some years ago by Ellen that they were working in CRISPR. So I was asked how this can help to CRISPR, but not to nuclear transfer. I think that if you are doing nuclear transfer, after this, maybe one of the regular policies is to do PGTA to be sure that everything is okay. I don't know. I cannot see the relationship. If you do nuclear transfer and after nuclear transfer, you want to test that all the chromosomes are there and there is no missing chromosome, you can do PGTA or you can do non-invasive PGTA. Yeah, I will say that it applies in the same way that it would apply the PGTA.
Okay.
Because I remember many years ago, the very first attempts, I think it was just going of doing a cytoplasmic transfer to have like renewal of mitochondria in all their patients. They stopped that, and they have several cases with monosomy X. So I guess that every time you do a manipulation, you need to be like cautious relating to the possibility of creating an aneuploidy. So in this sense, it makes sense.
The procedure in itself is a bit complicated. It's a complex. So is NIPGT a method that might replace PGT in the future? I think
I like to be cautious. I say that step by step. What we have seen now, we didn't want it to say, okay, this is right, because I think that we can improve the technology, and the technology should improve in the same way that PGTA has improved. PGTA was not the same 20 years ago. I think now we are in the phase of saying, if a center, as I mentioned, this one in Poland, they are having very good results. They feel comfortable with the results. They are doing this shift. They are changing PGTA to non-invasive. I will say every center to start little by little to see the results. And I will say explain everything very well to the patients. Explain before the treatment to the patient. We can do chromosomal testing to your embryos. You have these options, and explain the pros and cons. And I will try to make the shift smoothly. I think that for good prognosis patients, we are right there. For older patients, I will explain to the patients the two options.
Right. Okay. Yeah, Carmen, just coming to the same point, so many doctors I really see so much work has been done on non-invasive PGTA. So we have so many papers, publications available that justify the non-PGTA, non-invasive PGTA. But still, the doctors, they have a they are still have a fear of going about the sensitivity and specificity of non-invasive PGTA. They still they are not very comfortable with this thing. So do you have anything to say for those doctors, especially clinicians?
Right. We had a question, something on similar misconceptions of NIPHT or PHTA. So maybe that's something relates to this.
Yeah, I would say that if they don't feel comfortable with the sensitivity of the non-invasive, they should stay with the trophectomy biopsy. Because I think we are going to have variability. What we are observing now is some variability among centers depending on how you culture the embryos, how you collect the culture media. It's not the technique, it's the quality of the sample that is collected. So if a patient wants just a screening and priority, go to the non-invasive PGTA. The same thing with prenatal. Prenatal, non-invasive prenatal testing is a screening. If you want to be 100% sure, still the standard, the gold standard is PGTA. But said that, many clinicians are moving to the non-invasive after starting to apply that. But I want not to tell you a different thing. There is going to be a moment, and it's going to be, in, I would say, in a couple of years or so, in which we will be able, both in the non-invasive prenatal testing and in the culture media, to identify better the presence of maternal contamination in a cost-efficient way. We can do now with methylation, but it's very expensive. So you will tell us, I'm not going to pay that because it's going to be very expensive. But we are going to have something for sure in prenatal and in preimplantation to be able to be more sure that what we are analyzing is really the embryonic DNA. And at this point, we will move from screening to diagnosis. Once we will reach this, we can apply this to translocations, to monogenic diseases, and this is coming. I think that now we are at the screening part, very close to diagnosis for aneuploidy, and in the future, we will go to the diagnosis phase for translocation for monogenics.
I think we know about your also studies which you are doing, I think, on the maternal blood with regards to cytogenetic testing and other things. I think Igenomix is involved in cell-free DNA testing for the maternal misabortion cases also, right? And that is also on the diagnosis front. And if you club with NIPGT, it's going to be mind-blowing, definitely.
Yeah, we have been testing many different options, and as you can imagine, we are working on that. Also in at the prenatal level, because we also have a team of non-invasive prenatal testing. So we are trying to find cost-efficient technologies, because what we have now is too expensive for regular application. Right. And what is working very nicely now is the possibility of adding a blank. So every time you have a non-invasive PGTA case, you should send also a tube with culture media that has not been in contact with the embryo, but has been in the same dish and gone under the same procedure. And this is allowing us to identify at least the external contamination, and it's been very helpful. And this is cost efficient. So this is not so. Yes. So this is the challenge. The technology is there, but to make something cost efficient is the challenge.
Great. Thank you. So that also is something which we can have as future of genetic testing or future of IVF.
Just as intuitively, I'm asking as a rapid fire, one question, single question. Would you call NIPGT, invasive method, as a gold standard for embryonic screening?
I think it's for many people is now the gold standard, but I would say that 5 years, I think biopsies are going to decrease a lot. Okay. I know that this worries to some embryologists that they are very experts in biopsies. So I think I could say we will not. I think mainly in the States, in which they do a lot of PGTA, I would say no zero biopsies, but we will be decreasing the number of biopsies in the next 5 years. I think it's not going to be very fast, because what we have learned, we launched the non-invasive, how long it was, more than 3 years ago, and the adoptance of the technology is growing little by little. So like be relaxed, because I foresee this decrease, but it's going to be slow.
And another rapid fire question. So can we expect PGT, SR and M also included in the near future to be included with as a non-invasive PGTA method?
Yeah, I think yes. Some some people is very interesting. We were approached by the University of Brussels, that you know that they were like the ones that started with PGTM. They are interested in doing a collaboration. So we are working on that, but before we need to find the cost-effective tool to identify contamination. We are not going to move to monogenics or translocations if we don't have a safe and cost-efficient way of discarding contamination. I think this is key.
Okay. Next question. Should low mosaic embryo be mixed with a euploid embryo for a dual embryo transfer? Can we do this? Although I know that it's a no, but given a chance that yes, if the patient is insisting that, okay, I want to go for a dual embryo transfer, and they insist to go for so what is the impact of, basically my question is, what is the impact of mixing a euploid with a low mosaic embryo for a transfer? Single.
Okay, you are talking now about PGTA, right?
Right, PGTA. It's PG by euploid, yes, PGTA, basically.
Well, if it is PGTA, I think that you have to consider many things. If you have a mosaic embryo, I will look at the number of chromosomes that are mosaic. I will look at which chromosome is mosaic, and I will look if it is the full chromosome or just a segment of the chromosome. Right. And the level of mosaicism. If I have something like 40, 50% of mosaicism, I would, but this is my personal opinion. This is not science, it's my personal opinion. I will transfer first the euploid, and if the patient doesn't get pregnant in the first transfer, I will transfer the second. If it is a low mosaic, 20%, below 30%, I will consider this as a euploid. Everything below 30%, we consider as euploid. But again, and if you have many mosaics, then I will consider to do a rebiopsy, because if you have many mosaics, then I think it's noise.
Okay. So basically, it is obviously it should be the euploid only or mosaic only, and if at all we need to transfer, then you need to see what type of mosaicism it is, whether it is full mosaic or a partial mosaic, and which chromosome is involved. So that also definitely means a lot.
Mm-hmm.
So yeah, right. With regards to this mosaicism, we also have a question in the chat, and that is that could you explain the difference between high mosaic embryo and a complex mosaic embryo, which is reported in report many times? Okay. High mosaic embryo in most of the genetic reference labs is considered about 50% of abnormal cells. So something between 50-70% we consider high mosaic, and about 70% is aneuploid. And what is very clear from many publications from us and from others is that about 50%, the behavior is very similar to the completely aneuploid embryo. So a high mosaic embryo is kind of a variation of an aneuploid embryo. And a complex mosaic is an embryo that has mosaicism for more than one chromosome. So if you have only one chromosome that is mosaic, this is mosaic. And if you have 2, 3, 4, 5, this is complex. If it is an Igenomix report, everything above 5, we consider chaotic, because chaotic is like a chaos. So when you have a sample from a culture media, from a biopsy with 6, 7, 8, 9 abnormal chromosomes, this is in most of the cases showing that this is a damaged degenerated DNA. So this is a chaos. Like a blastocyst, a human blastocyst cannot survive to blastocyst stage with so many abnormalities. Imagine the cell divisions with this chaos of chromosomes. So most of the chaotic embryos on blastocyst biopsies are artifacts. That's why one of Igenomix recommendations is, if you have a chaotic embryo, consider
kind of non-informative. You can rescue, you can try to rescue this, and in more than 50% of the cases, you can rescue it.
Okay. Right, so we have the difference now. If it is in one embryo, it's basically, if it's in one of the chromosome, it is high mosaic. If it is in multiple, it can be complex. And if it is, and if it is many, then you term it as chaotic.
If it is more than 10,
then
it is chaotic, and between 50 to 70%, that is high mosaic.
Exactly.
How much do you believe in, you were mentioning while answering with regards to low mosaic embryos being the second priority, but then how much is the belief in self-correction abilities of these low mosaic embryos? Is it just something which is theoretical, or is it something which has a real scientific impact?
Okay, wow, this is interesting. Talking about, I don't believe about self-correction of aneuploidies. I believe about embryo correction. I believe that the embryo is really smart. The embryo is really smart, and we know that embryos carrying certain abnormalities, they don't progress to blastocyst stage, and they arrest before. So we know that haploid embryos or triploid embryos, they arrest in many cases, not always, we know. We know that embryos with monosomies arrest more than embryos with trisomies. And if you have multiple aneuploidies, whole chromosome aneuploidies, they arrest. So embryos with 5, 6 aneuploidies, usually they don't reach blastocyst stage. And we have learned this by comparing the results of day 3 biopsies and blastocyst biopsies. When we were doing day 3 biopsies, at that time we had 15% of chaotic embryos. That means embryos with 6 or more chromosomal abnormalities, 15% on day 3. When we move to day 5, day 6 biopsies, the percentage is 2-3%. What has happened in the middle? They have arrested. They have arrested. The ones that most of the embryos that they don't reach blastocyst stage is because they are chromosomally chaotic. Then at blastocyst stage, we have 2 or 3%. Few of them are going to be real chaotics, but maybe 6, 8 abnormalities. And a part of them are going to be artifacts because of the generated DNA. That usually is when the cells are lysed with the laser. If you don't have intact cells, usually you have very noisy profiles. So I believe in embryo correction. I don't believe so much in self-correction. We know that we have from pregnancies, we have the fetoplacental discrepancies that can be about 1 to 3%. So I can understand that like inner cell mass trophectoden discrepancy of about 3%. I think this is physiologic, and this is what I can understand about this. And the same correction appears after implantation. When we are studying miscarriages, we don't have miscarriages with more than 3 aneuploidies. You always observe, if you analyze the product of conception and you do karyotypes or you do NGS analysis, aneuploidy for 1, very rarely 2, very rarely monosomies. So we know that this is this selection in the development more than the chromosomes.
I'll remember that. It's not self-correction, it's embryo correction. Embryos are smart. It sums it up.
but this is Carmen's theory. This is Carmen's theory.
So maybe we can take the last question.
Right. So there's a last question. Again, I suppose it's more related to based on mole biology, and that thoughts on MLBAC versus reprosequencing versus Supralex for NIPGTA. Thank you.
What can you repeat?
Supralex. So you can just read it in chat. I think it's thoughts on MALBAC versus Repros-SEQ or Sureplex, SUREPLEX, Sureplex for NIPGTA.
Okay. Well, I can tell you we have test Malvac, we have test Reprosec, we have the sureplex, we have the very sick, and we have the embryo map. And I think there are no big differences among them in the enzyme, but you have to, ¿cómo se dice? I don't know how to say in English. You have to trick, can you say that? You have to trick the protocol. You cannot use the standard protocol you use for the trophectoden biopsies to apply to the culture media. For example, we don't do the lysis. In most of the protocols, we remove the lysis steps. And also in some protocols, FreeProsec or sureplex that you have pre-amplification and amplification, we trick the thermocycler protocol to have more cycles, different length. So this is the kind of things that we do. So you can work with them, but you need to work in the protocol, not just use the commercial protocol easily. And the very, very, very important thing, one thing is the protocol. The very, very important thing is to identify the number of reads that you get. It's very important to understand the sequencing depth and then how you analyze the results. As I mentioned, the way of diagnosing is different. No, you cannot apply the commercial software to this, saying everything below or below 30, 20 is normal, everything above this is aneuploid, the same way of reporting mosaicism. If you apply the commercial softwares, then you are going to have most of your cases having mosaics or being abnormal. I think this is the big difference.
Okay, good. I think so. We need to have maybe another session on the details of how analysis has been conducted from the point of view of who is actually working on the sequencer and analyzer, because that throws a lot of light on the science actually going behind the reporting, which possibly is not known by the common mass. And if that has been known by embryologists, I think they will, or doctors, it will be very effective for them to take a consented decisions.
Yeah, so probably will also require a clinician.
Yes.
Right?
Yeah, I think it's very important to consider one thing if you are testing how to work on this. So imagine if you have 50% of aneuploidies in PGTA in a 35-year-old patient, if you do any PGTA, the incidence of aneuploidy should be similar. You cannot have higher incidence of aneuploidy in the same type of patients in the culture media. Then something is failing. You have, this is an easy way of comparing that. And I suggest you to do that. Take patients with the same characteristics and compare what are your results with PGTA and with Embrace in terms of aneuploidies. And also aneuploidy types. You cannot increase the rate of like trisomies for sex chromosomes. This is not real in the culture media, for example. There are some abnormalities that we know that they are artifacts. Duplications, small duplications in the culture media. We consider this benign because we know that they are artifacts due to the size of the small fragments. So these kind of things, you gain knowledge by the years and by comparing with trophectoden biopsy. So these are important things just to understand your rates.
So I think with this, we are through with the questions, and also it's as the time is approaching to say goodbye, although it's very hard to say goodbye, we were really enjoying the interesting talk. And the conversation was so interesting. And before leaving, I would like to thank Igenomix who were courteous enough to arrange this webinar session in such a short duration, and especially to you for taking out time out of your busy schedule and giving us time to share your experience and thoughts on non-invasive PGTA. And Carmen, so with this, I would really like to thank from Indira IVF family as well as from IFA, Indira Fertility Academy, which is an academic wing of Indira IVF. And here we, as I told you, we are providing short-term as well as long-term courses And we do have a course on biopsy. It's a 7-day course, and we have a course on, we give extensive hands-on on biopsy procedures. So if anybody is interested, you can just contact us. You can just drop us an email or call us. We are right away, and we'll definitely do the needful.
Right. It's always exciting to converse with Dr. Carmen. I remember the question and answer session of our previous talks also. The way she simplifies and answers and understands the questions and answers, it's really very insightful. So thank you very much for that. And thank you for your time. And thank you, audience, for being such a lively audience, asking questions and making this session all the more interesting. Thank you very much.
Thank you.
Thank you. Thank you. I have really enjoyed the session, and congratulations for your academy efforts for training.
Thank you. Thank you.
Thank you.
Thank you, and have a nice day.
You too. Bye-bye.